So last week we finished off talking about G protein receptor signaling and the different ways that those cascades work. And we ended our lecture talking about how G proteins can also regulate not just other proteins and enzymes in general, but also specific channels, like ion channels that are important in cell signaling all the time. And we talked about how it can do that by activating other proteins that are G proteins, like and other messengers that can, like the inositol triphosphate and diacylglycerols, that can be good quick messengers for our cell signaling pathways. And then we saw how in an egg, once a sperm fertilizes the egg, one of the first things that happen is this influx of iron, zinc, and calcium in the cytosol that will make sure that it becomes impermeable so that no other sperm gets in, and it's now locked up essentially for developmental process. And so that was where we ended, and I had the link for that video for you there. So today we are going to now move on, and we are going to first talk about another kind of messenger that can get activated, so-called from G protein. So G proteins not only use ions and other small molecules as second messengers, but they can actually use the byproduct of their reactions as second messengers. So if you think about adenosine monophosphate, or AMP, all that is broken down ATP, right? The phosphates have been released, and now you have the AMP left over. And that AMP can be a very potent second messenger in itself. So in this case, what is happening is that the adenylyl cyclase is activated by the G protein itself, and then that is breaking down ATP like it does for energy use. And the remaining AMP that is left behind is what is going to be then acting as a second messenger. Um, Now, this AMP is a little bit different than your typical AMP, right? In this case, you not only have those two phosphates, the pyrophosphates, removed from ATP, but you also have cleavage of this remaining phosphate to create this cyclic AMP structure. Now, one of the biggest things that this CMP or cyclic AMP targets is, different protein kinase structures. Um, so our protein kinases. Now, protein kinases' job is to phosphorylate other proteins and make them either activated or inactivated, depending on whatever function they need to perform. The cyclic AMP binds to particular structures inside the cytoplasm, where their main job is to activate the protein kinase molecules. So here you have different regulatory proteins that can be bound to, that have binding sites for cyclic AMP. These proteins are usually going to be bound to catalytic subunits of protein kinase. And when cyclic AMP is bound to them, that binding is disrupted. So now your catalytic subunits of those protein kinase A are going to be activated so that they can go and phosphorylate their target genes, target proteins, to regulate gene expression. So here you can see it a little bit more broadly. You have your little cyclic AMP molecules. They are going to bind to the catalytic units in the protein kinase A, activating them in response. Those active protein kinase A will usually go through the nuclear pore into the nucleus, where they bind to inactive transcriptional regulators. Those, when activated or phosphorylated, will bind to the actual gene sequences that are not gene sequences, DNA sequences for that particular regulator, and activate target gene expression. this reminds me of the question that I was thinking about. So there was that question about repetitive sequence in the DNA. There are many different types of repetitive sequences in the DNA, some that we've talked about and some that we have not talked about. So what I was looking for were actually sequences that we have talked about. You did get partial credit if you talked about those tandem repeats or repeats where it leads to Huntington's or muscular dystrophy and whatever. But what I was looking for in specific for that question were repetitive repeats like the one seen in telomerase or telomeres of our cells or chromosomes, or the TATA binding box, right, in the promoter sequences. The specific sequences like the one for NANOG to bind these transcriptional regulators. I was looking for these specific sequences, not the actual sequence, but that idea. That's what I was looking for that question. That's what these, this is a way for us to regulate how many different regulators or how many different proteins or master regulators we need inside our cells. We don't need individual one for every different gene because there are these repetitive sequences that allow the same regulator to bind to multiple genes and activate them. Does that make sense? Yes? No? Do you guys know which question I'm talking about?
Yeah.
Yes. Yeah.
Okay, so now a lot of our responses in our body are some of our actual, responses to environmental stimuli are regulated by or mediated rather by cyclic AMP. So it's giving you a few of those examples that you should know about. So you have these molecules, right? Extracellular signaling molecules that are binding to the actual plasma membrane receptors or coming into the cytoplasm and binding to those. These are binding to G protein receptors, epinephrine, glucagon, or ACTH, which is a hormone. And they are going to, in their target tissues, have a major response to that environmental stimuli. So in the heart, epinephrine will increase the heart rate and how forcefully the contraction is happening in the heart at each heartbeat. However, in the skeletal muscle, it's going to break down the glycogen. That's the storage of energy in your muscle fibers, right? As a response to this extreme high epinephrine level or activation of epinephrine level. And then the adrenocorticotropic hormone is going to, these are all in response to kind of fight or flight type of responses. And this would lead to cortisol secretion, which is a hormone that is increased when you have high levels of stress by the adrenal gland. Okay. And all of these responses require cyclic AMP as that mediator, the second messenger in that G protein coupled receptor signaling. So here you are going to see that in a little bit more detail. So when an epinephrine molecule binds to the G protein receptor on the plasma membrane for epinephrine, these specific ones are called adrenergic receptors. And this particular one is going to activate the G protein, which will then break down ATP to cyclic AMP. That will activate the protein kinase that leads to glycogen breakdown as the downstream pathway that is the response. So that's one way that it is affecting, causing a very quick response. So while some G protein coupled signaling pathways are kind of slow pathways, anytime that you use cyclic AMP, you usually end up with a very fast response. So we talked about how, you can have these fast responses versus slow responses. In slow responses, you're usually affecting gene expression. However, when you're using cyclic AMP as a second messenger in this type of pathway, you are using this to activate this super fast response by simply activating a protein that's already there in the form of protein kinase that can then lead to a quick response to that environmental stimulus. Another final thing that can also be affected or that can also interact with G protein coupled receptors are just some dissolved gases. And these, again, it can include some nitrous oxide or even carbon dioxide in our system that can easily go through the plasma membrane itself. But these can also bind to the actual G protein receptor and form a response. So, one of the things that it can do is that other molecules, so there are two ways that it can have G protein coupled receptor signaling response. One is obviously the if you were looking in our blood vessels, as you have the gas exchange happening, you will have these gas molecules that can bind to the G protein receptor itself. But also sometimes you have these molecules like acetylcholine, actual signaling molecules that are binding to the G protein coupled receptor. And in response to them, you will have either nitrous oxide or carbon dioxide having rapid diffusion across the membrane from the blood vessels to the cells or vice versa. And that leads to a response in the target cell. In this case, when we're looking at the nitrous oxide going from the endothelial cell, so inside our blood vessel, into the muscle cell, it will lead to a relaxation of the smooth muscle cells and it will act as a muscle relaxant in that case. So this again is to remind you how G protein coupled receptors can trigger intracellular signaling cascades, where essentially you are activating, not just having an activation of the signal, but also you're enhancing the signal, right? You are kind of multiplying its activity each time at each level so that you can have super high sensitivity and you can change the cell. You can get that response in a very quick and effective manner. So here you have an example of a rhodopsin molecule that is getting absorbed, that is absorbing light from outside. And it is in a very few steps, it not only activates that pathway, but enhances it so that our signal that is relayed to the brain can detect that light and respond to it, right? So this is something that you would see in our photoreceptor cells in our eyes, and how quickly they can respond to super bright light, for example, or just something that they are visualizing in the environment. Was there a question in the chat?
No.
And okay, so one thing to notice here is how a single, rhodopsin molecule getting activated is leading to several proteins, right? Over 500 G proteins getting activated. And those 500 G proteins are going to release 500 cyclic GMP phosphodiesterase molecules that are then, each one of them is going to activate multiple GMP molecules, right? Not a single one. So you magnify that response. And then each one of those is going to lead to certain channels getting opened, and then the response happening at the end of the day to signal the brain about that light. Okay, so now switching over to a second type of receptor that is present in our membranes. These are receptor tyrosine kinase, which are also going to be coupled with enzymes very similar to G protein in that way. However, the receptor tyrosine kinases are different in that they themselves are going to activate downstream intracellular signaling pathways by activating multiple second or signaling molecules themselves. So in G protein just transferred the signal to another molecule, one single molecule that then went and activated other molecules. Here, a single receptor tyrosine kinase can activate multiple molecules that each one of them will then activate intracellular signaling proteins. And they do that by having many, many phosphorylation sites on their intracellular domain. So once receptor tyrosine kinase is activated by uh, binding of a signaling molecule on the outside, they work as dimers. So those two domains come together and the kinase activity of that particular receptor tyrosine kinase is activated. Now, once that is activated, multiple phosphorylations will happen all along various amino acid residues in that intracellular site. And each one of these phosphorylations can activate a different intracellular signaling protein. Now, depending upon the type of the cell you're looking at or the environmental stimuli you receive, sometimes all of them may be activated. Other times, maybe just one or two of them are going to be activated. But the phosphorylations that happen along that intracellular arm regulate which intracellular activated molecules are going to occur from there. And then those pathways are going to be activated downstream. So looking at that a little bit more closely, here is an example looking at two different receptors that are receptor tyrosine kinase. One is the PDGF beta receptor, and then the epidermal growth factor receptor. These are both actually growth factor receptors. Now, looking at these, you see that there are multiple residues all along the intracellular arm that have different phosphorylation sites. And each one of them is going to be activating in different protein molecule, right? Sometimes both like the 1086 and the 845 residue phosphorylation activates the STAT3/5, right? But most of the times, each one of these phosphorylation is specifically targeting a specific intracellular downstream pathway. So depending on which one of these are getting phosphorylated in a particular system, that downstream pathway is going to be activated. So by doing this, a single receptor tyrosine kinase can make hundreds of different versions of activations based on how many different downstream molecules they regulate. Okay. Now, sometimes these receptor tyrosine kinases will activate G proteins as one of their secondary responses. So here you have a molecule where once activated, one of the phosphorylations will bind to an adapter protein that can then bind to a G protein factor, exchange factor, and lead to that activation of a inactive G protein to an activated G protein. So in this case, even though this protein can also be activated by a G protein receptor, this can also be activated, it can be activated two different ways depending on its environment. It can either be activated by its G protein receptor, or it can be activated by a receptor tyrosine kinase, which then is activating it through the G protein exchange factor, right? So it is making the inactive RAS protein into a activated RAS protein. Now, this usually happens for growth factors. A lot of receptor tyrosine kinases are growth factor receptors, and they activate downstream growth factors or growth factor related pathways. So RAS is a very potent mitogenic response. It activates the growth signaling pathway that leads to the cell replicating and dividing at a really high rate. So that, like I said, can be activated two different ways, and receptor tyrosine kinase can activate some of these monomeric G proteins as well. Questions on this? Does everyone get that? Yes? No? So how do these activate the growth response? Is through activation of downstream pathways, obviously. So once the RAS protein is activated, which is our G protein that got activated by the receptor tyrosine kinase, it will activate downstream growth signaling pathways like MAP kinase pathway. Again, MAP kinase pathway is a very potent growth pathway. It has a lot of mitogenic signals that will activate growth response. And it works both through slow as well as fast mechanism. Some of it works through simple activation of other mitogenic proteins that can then promote, for example, shift of cells from a G0 into synthesis DNA replication phase or into cell division phase. And other parts of the kinase pathway are transcriptional regulators, which are going to regulate gene expression and activate genes related to growth and cell cycle control. Another important pathway that receptor tyrosine kinases activate are other kinases. So, for example, PI3 kinase or Akt pathway. This again is going to use an inositol phospholipid as a secondary messenger or mediator. So in this case, the Akt pathway, so MAP kinase pathway is a growth stimulating pathway, right? Akt pathway, on the other hand, is a survival pathway. It's a way for the cell to say, oh, there is something stressful in the environment. There is something damaging in the environment. How do I protect myself? How do I survive and instead not die in response to it? In this case, you get a signal, again, bound to a receptor tyrosine kinase that will prevent death from happening in this stressed cell, and that will activate the receptor tyrosine kinase. This particular receptor tyrosine kinase will phosphorylate PI3 kinase that can phosphorylate these inositol phospholipids that act as second messengers. These phosphorylated inositol phospholipids are then going to bind other protein kinases and activate those, leading to activation of Akt pathway, which then um, leads to activation of survival signals downstream of it. Again, there are going to be several steps in that pathway further on as well. One of the pathways that it activates, or one of the proteins that it activates for survival, is the bad Bcl2 complex. Now, normally, the bad protein is bound to the Bcl2 so that it is unable to work. Bcl2 is basically inactivated and it cannot work. When there is a survival signal that leads to activation of Akt, it phosphorylates the bad protein so that it can no longer bind to the Bcl2. When Bcl2 is not bound, it is active to do other work. And one of the works that it does is promote cell survival by inhibiting apoptosis. And it does so in a very special way, which is related to, again, some of the work you guys are doing in your lab this week. Remember caspase 3? Yes, no, maybe? What's caspase 3? Huh?
Used in the apoptosis of cell program death.
Yes, programmed cell death and apoptosis. So one of the way caspase 3 is activated is through this internal apoptotic signaling pathway that leads to release of cytochrome C, a small molecule from the mitochondria. Well, what Bcl2, activated Bcl2 does is it binds to that cytochrome C, so it can't be released. If it's not released, it won't cause the apoptosis because it can't upregulate, it can't activate that process. So that's one of the ways that it promotes survival of cells and inhibits apoptosis. This pathway is many times activated or enhanced in cancer cells. And that's one way cancer cells can protect themselves from, all these toxins that are around them, or all these, if that's one of the ways they can become resistant to drug therapy, is by activating this all the time, Bcl2 all the time, so that it's bound to cytochrome C. It doesn't allow for its release, so apoptosis cannot happen. Okay. Now, this is another way that, it's basically showing you how Akt can also promote cell survival through non-Bcl2 ways. One of these is by, so this is, looking at activation of TOR, which is called target. It's short for target of rapamycin. They named this protein, probably some very boring scientist named it, other than the ones that named them cool ways, because rapamycin is an antibiotic that was given to kill some certain types of infection. And this particular receptor protein is the target of rapamycin drug. And so that's why it's called that. It is also used as an anti-cancer drug now that we know that it can act in this survival pathway. But rapamycin is, well, TAR, or target of rapamycin, that works two different ways in the survival pathway. It can inhibit protein degradation, which is again one part of general, toxicity response. If there are lots of proteins that are not folded properly or that are damaged, they're going to get targeted for degradation. But also, proteins get targeted for degradation during a normal apoptotic response. That's one of the things caspase 3 does, is targets proteins for degradation to kill them so that there are no proteins for the cells to work with and the cell will eventually die. Well, TAR, once it's activated, can inhibit that by inhibiting protein degradation. It can deubiquitinate proteins and it can enhance protein synthesis, so there are more proteins getting made and less proteins getting degraded. It can also, so it's doing that too. It can inhibit protein degradation, but it can also enhance protein synthesis. Well, rapamycin can be used as an anti-cancer drug because not only does it act as an antibiotic, but in normal cells, it can bind to that activated TOR. Like if you have a cancer cell that has activated TOR or this target of rapamycin, rapamycin can bind to it and then allow the cells to target proteins for degradation and kill the cell through that mechanism. So normally in our body, TOR is not going to be activated if our cells are healthy. It's only going to be activated either in damaged cells or it's going to be activated in cancer cells as a survival response. That's why. Having said that, it's not to say that it can't inhibit those normal cells that were going through something similar as well. Okay. So now I think you can appreciate from some of these things that we've just talked about that many of these different receptors and many of these signaling pathways, they're interconnected, right? They're not all just G protein coupled receptor signaling or receptor tyrosine kinase signaling or a single transcription factor working alone. These pathways are highly linked inside our cells and they're cross-talking with each other constantly to maintain the equilibrium that we have. So you have simple G protein coupled signaling, but you also have G protein coupled signaling where in these are these different receptor molecule. Am I breaking up?
Yeah, you froze for a good bit there.
It's back now.
What's the last thing you heard? What was the last thing you guys heard?
Just to start from this slide, the beginning of this slide.
Basically what I talked about is that, I think you can appreciate from the slides before that these different receptors are not working alone. They are working in crosstalk with each other, in collaboration with each other. And so while there are some signaling pathways, they are just activated by G protein coupled receptors or just by receptor tyrosine kinase. There are way more where they are kind of talking with each other and interacting with each other at various levels, throughout the pathways to give you the final response that you look at, whether it is transcriptional regulation or just simple regulation of proteins, activation, deactivation, to get the response you want, right? And so we need to be mindful of that when we are looking at a pathway to see how it's going to create the response at the end of the day. Now, some receptors activate a protein that is a fast track to transcriptional regulation, leading to a little bit faster response than a typical transcriptional regulation. The Notch receptor itself is one of those things where that receptor that was outside on the plasma membrane can directly stimulate a transcriptional response by binding to a signaling protein called delta, which is usually on another plasma membrane of the nerve cell. When they are bound, portion of the Notch protein gets cleaved. So that receptor, part of it just gets cleaved or chopped off. And that cleaved receptor itself is a transcriptional regulator. So it is bypassing all those mini steps in between, and it's going straight to the source and activating a response much faster than it would if it was using a signaling cascade, right? So in this case, you have the receptor itself do that. Caspase 3, for example, or most of the caspases in our body, they are present in the body all the time at some basal level in our cells. When they're activated, they get cleaved, and that cleaved portion is what's activating the response. So many proteins have that pathway, that type of pathway in them, where the protein itself, once activated, is cleaved and a portion of it goes off and does the response that we wanted to look at. Okay. So again, this is to remind you different ways that intracellular signaling can happen from outside signals being received on the receptors. You can have a signaling pathway where the signal binds to the receptor outside, then activates a kinase, like a receptor tyrosine kinase, that will then activate other target proteins and cause a cell response. You can have pathways where, like the G protein coupled receptors, where the signaling pathway, signaling molecule, when bound to the receptor, will activate. Well, this is actually again a receptor tyrosine kinase, where multiple kinases that will each then activate their own target proteins and elicit responses. Or you can have G protein coupled receptors where the initial pathway will bind to a catalytic domain of another protein, activate it, and have a response right away. Then you can also have signaling molecules that can inhibit activities of kinases. So their job is not to activate, but inhibit the response that you may have gotten from another molecule, right? So there are many different ways that signaling molecules can affect our cell response. They don't always have to be activation of signaling or activation of kinases. They can also be there as inhibitory responses as well. Okay, so that's all for this one. Questions about cell signaling before we go to more cell signaling in the form of cell cycle, cancer biology? Yes, no, maybe? Leslie seems worried.
Not currently.
You're good?
No questions currently. Don't know what to ask just yet.
Sounds good. So let's go straight into cancer. Now, I don't know if my cancer bio lecture on the YouTube channel is as elaborate. I do have a mini version of this on the YouTube channel that I can link as well, because I know we won't get done with all of it. But I do think that Dr. Orloff's lecture is amazing. So you can just listen to him and get a break from me. Has anyone seen that lecture yet? The one from Dr. Orloff? Oh, gosh, sorry, I'm excited about it all the time. Okay, so we are going to talk about cancer. Why are we going to talk about cancer? Anyone?
Many people suffer from it.
Yes, many people do suffer from it, but why now? Why we are talking about it in molecular and cellular biology?
Functions of the cell, and now we're digging into what happens as a result of different proteins activated.
Yeah, so we just talked about cell signaling and how it controls the different responses in our body. Well, cancer basically is when a cell doesn't listen to its signaling pathways anymore, and a step is broken down somewhere in there leading to an aberrant response. So it's a cell not following the rule anymore, right? The rule of the land. And that's what leads to that response. Why do I have some mummy fingers on there in the corner? She's like, ooh, these are mummy fingers? Fingernails. I don't know if he died from cancer, but they did get cancer at that time as well. So it isn't a new disease. It's happened for a very, very, very long time. Having said that, I have a dinosaur there too. Did he die from cancer too?
Maybe.
Maybe. Uh, yeah, they're not going to be living cells, but we can look at them and we can see stuff in them and evidence of different diseases in them. Yes, dinosaurs got cancer as well. We've seen it in the dinosaur fossil bones. We can tell that they had those. But humans, you can see recorded history of cancer from a very long time, surprisingly long time, describing cancers all the way back to 3000 BC. We can also see evidence of cancer in these fossilized bones and human mummies from ancient Egypt, as well as from South America, and read about it in their manuscripts. Um, so they reveal that they got all kinds of different types of cancer, but one of the most common ones seemed to be the bone cancer called osteosarcoma, and also head and neck cancer or oral cancers, possibly because of certain dietary agents that they were exposed to or that they ate, like the betel nut or some of the other things that we now know can cause cancer. Um, the first primary literature article, primary research article for cancer is back in 1600 BC. That's it. Um, and that has been, description. Well, there are some documents all the way back to 3000 BC, but there's an actual study around 1600 BC that describes 8 cases of tumors that they tried to treat with a process called, well, they called it fire drill, but it is essentially cauterization of the bloodstream. And while they showed that there was, some alleviation of symptoms and a moderate stall of growth for some period of time, eventually those grew back, and they said there was no treatment. How do you think that using cauterization would have had any effect at all at the tumor like they saw? What happens when you cauterize around that tumor mass? What are you doing?
You're cutting off the blood supply to that area.
Yeah, you're basically cutting off the blood supply. And you're not really killing those cells that much, but what you're doing is that you're cutting off the blood supply. When they don't get any nutrition or oxygen, just like normal cells, these are cells. They are actual living cells. They require oxygen, they require nutrition. And if they don't get it, they will die. So that's what they saw with their cauterization methods, which probably weren't complete or fully able to do the way they wanted to. They didn't get all the capillaries and everything. But they saw that initial response of shrinkage. However, because it wasn't complete, they probably ended up getting them back. At the same time, if you have a tumor mass that's anything more than 1 centimeter, even if you were to remove 99.9% of it, you still have thousands of cells that are circulating in your bloodstream at that point that are also cancerous. So eventually it would come back unless you got rid of them in some other way. So these are statistics from 2018, so already a little bit outdated, but over 17 million cases are come out of cancer every year, and nearly 10 million people die worldwide each year from cancer. So it is a major health burden, right? Major health crisis that is something that we go through every year and does need to be improved for sure. About 40% of people at this time, they suspect will be diagnosed with cancer at some point in their life. That's a really high percentage if you think about it. Yes, many of them will be able to receive treatment that can lead to even cure, if not just alleviation of symptoms. But those treatments are very slow in getting updated, and there is a need to get those treatments out quicker and to get them better developed as well. So how do these cancers happen? Well, most of these cancers are not actually genetic disorders. Only about 1 to 5% of cancers, depending on the type, happen because of genetic history for that cancer. Majority of our cancers are through somatic cell mutations. That means it's just our normal cells that over time, as they're replicating and dividing and getting exposed to environmental stimuli, are getting these mutations that make them more likely to have an essential pathway broken down. And as those essential pathways break down, they'll lead to aberrant cell growth, and all those mutations lead to the genetic instability that is the hallmark of cancers. And one of the ways that we know that is because we see that as the age increases, you see higher rate of cancers happening in individuals across the board, regardless of where they come from. So the other thing with the tumors is that tumors are curable if you catch them early. Unfortunately, most tumors are not going to be diagnosed until they are visible, right, or palpable, which means that there are thousands, hundreds of thousands of cells, if not millions of cells, that are not just part of the tumor, but also circulating in your bloodstream at that point, because the cancer has had the chance to grow and mutate and evolve into a more aggressive phenotype. If you have ways to pre-diagnose cancer, for example, with mammography, breast cancer, we have seen that we can usually catch them a lot earlier, right? Because they're visible on those screening methods a lot earlier, which helps manage them better and gets you higher survival rate. And like I said, once the tumor becomes actually palpable and visible, there are so many cells in there that you've already gone through that large growth curve, and now you're at that point where it's hard to remove all of that from the body. So what causes it? Well, there are 3 different categories of carcinogens that can cause cancer. They can be physical, chemical, or biological carcinogens. Now, carcinogen is any agent that can cause a mutation that can lead to transformation of a normal cell into a cancerous or precancerous stage. Now, these mutations have to be in specific pathways, and we'll talk about those in just a few minutes. But basically, it has to break down one of those signaling pathways, right, at a critical point so that it's either no longer active or always active. Now, physical carcinogens are things that can directly break down the DNA and cause a mutation in the DNA itself directly. And examples would be UV or ionizing radiation that cause double strand or single strand breaks in the double helix. Chemical carcinogens are carcinogens where either they themselves or a byproduct of those compounds can then cause damage to the DNA and lead to mutations over time. Examples of those are chemicals such as asbestos, tobacco smoke, aflatoxin, which is seen in a lot of food contaminants and grains, for example, and arsenic, which is a common contaminant, or lead, for example, that can be a common contaminant in drinking water. And then finally, biological carcinogens would be bacterial and viral infections that, or even parasitic infections that can affect the DNA by either integrating into the genome or affecting the biology of that cell in a way that can lead you to a higher rate of cancer. Now, viruses, many times, you have some viruses where they can integrate their genome into your genome, and that's how they can cause a mutation. Bacterial infections, many times, what they do is not really, they're not changing the DNA directly, but what they're causing is an inflammation response in that system, whether it be, and many viral infections can work this way as well, whether it is HCV and liver or another virus like that. Those viral infections cause this microenvironment around, or bacteria can cause a microenvironment in the system where they are, where the cells are constantly inflamed and stressed. And that leads to a higher growth rate in these cells, for a higher turnover of those cells, which can lead to higher rate of mutations and higher chance of cancer. The more our cells replicate, the more chances there are that there'll be a mistake that will be uncaught that can lead to, again, mutations and cancer. Okay. So when we look at our, these individual categories, and we look at how that affect our systems, there have been epidemiological studies done for all of them and more all across the globe to identify preventable causes of cancer, which is why we know that tobacco use leads to cancer, and also that if it's stopped in use or if it is discouraged, you see the opposite trend, that the rate of cancer go down over time. And so here you can, for example, see that when you saw the high rate of cigarette consumption, about 20 to 25 years later, you started to see an increase in lung cancer incidence and death. And then once that rate started to go down, again, the similar trend appeared on the other end in the same amount of time, in a slightly less amount of time, but similar. Another example is looking at colon cancer rate cases per 100,000 women per year in various countries. And this is looking at meat consumption per person and how that relates to colon cancer. Now, specifically, meat consumption can sometimes increase the rate simply because the animal, whatever it has consumed, has also already affected its system. And some of the toxins that it consumes get lodged up in the fats inside the animal, and they then are transferred over to us when we consume them. So we get not only the exposure from our own contaminants around our environment, but also whatever we consume, whatever exposure those fruits, vegetables, and meat had is also going to come to us. So for example, if we consume a crop that had a fertilizer given to it, or insecticide applied to it that can cause cancer in humans, and we consume it without washing it or removing that toxin, then we are also getting affected by it indirectly as well. Because now when we consume it, it's going to affect ourselves as well. And so you see a similar rate with that. The more meat consumption overall that a person consumes, the higher the rate of colon cancer. Now, cancer cells can usually be spotted by how abnormal their DNA looks. And we can, that reflects one main hallmark of cancer called genetic instability. That basically means that once there's a mutation, you're more likely to get a second mutation, which builds up another chance of another mutation. So these cancer cells accumulate mutations at a higher rate. And so while a normal karyotype, in this case, when you look at it, it shows nice organized chromosomes, all 23 pairs looking very similar. Cancer karyotype will show a lot of abnormalities, where you have a lot of translocations, you have duplications of parts, you have different chromosomes showing up in a very different way. And that shows that genetic instability of that system. Now, going back to, well, we get mutations all the time, but we don't get cancer all the time, is because to get cancer, you have to have mutations in one of 5 key pathways. Those pathways include DNA repair pathways. Obviously, if one of the critical repair pathway is shut down, you're more likely to accumulate mistakes that were part of that pathway to be repaired, right? no repair happens, more mutations will occur. Cell division, if you have a regulatory gene that controls when a cell is going to divide and it's now mutated so that it cannot control it appropriately, you'll have higher rate of cell division leading to aberrant cell growth. Apoptosis, similarly, if you activate survival signals or inhibit apoptotic response, cells are not going to die when they are damaged, even though they should be dead. And that will lead to more mutated cells accumulating in your body, leading to a cancer response. And then the last two are more hallmarks of higher grade cancers, where they become de-differentiated. So they are no longer, a kidney cell is no longer just like a kidney cell. It's looking a little bit more embryonic, which means it can survive in more types of environment, which means it can now can travel to another place and metastasize that and take over a different organ. However, it can't just move by itself unless it loses its cell-cell contacts and cell communication abilities as well. Then it can now travel to another place and migrate and cause a metastatic tumor. So those are the major pathways that have to be altered in order to get a cancer to occur. Usually, again, a single mutation is not enough. Usually you take more than one mutation to have that. Ooh, that's cool. Okay, so we're gonna stop here. I will actually I will see if I can record a Canvas Studio mini lecture and post that on cancer biology as well. But next time when we come, we are gonna move on to DNA repair pathways and talk more about those in our next lecture. And so the remaining of these hallmarks of cancer, going to them in a little bit more detail, you will do on your own using that. And it will show you some examples again of cell signaling molecules that are affected in cancer cells and how that can change our body's response. Okay?
So the lecture that it's online, is it related to this one? Is it similar? Yep. Okay.
Oh, yeah. It is. It's very similar.
It's
going to give you a lot more interesting facts. I did a mini version of that.
That's good. Thank you.
You love it. So yeah, so the lecture on there, the introduction lecture, it's pretty much going to talk about all the stuff that I just talked about and give you a lot more information about it and cool information about it. Okay?
Right, thank you.
You're welcome. Take care. It's actually going to talk about the rest of the stuff too that I haven't gotten a chance to talk about. So you'll be able to get all the information that you need. Okay? Okay, guys. Take care. Bye-bye.